Welcome to the next installment of cBioPortal for beginners, a tutorial. And so for this video, we're going to focus on the popular Kaplan-Meier curves and how we can make them. In addition, we're going to also talk about how to make Lolliplots plots as well. So what you see in front of you is a Kaplan-Meier curve, and this is all within non-small cell lung cancer. And so you can see in purple, we have patients who are TP53 wild type. In green, we have patients who are TP53 mutant. And you can see clear separation in the curves, and you can see patients who have a TP53 mutation in non-small cell lung cancer have a worse overall survival, which is expected. In addition, this is a Lolliplots plot. So this is the TP53 gene, and as you can see, at every point, there's a mutation that's represented on the TP53 gene. And so this type of plot is very powerful because it's able to be a graphic showing the heterogeneity of types of mutations of TP53, and you can see it's pretty widespread in terms of which the different types of mutations you see in TP53. Specifically, this is also non-small cell lung cancer, but you can see the taller these bars are, the more represented these TP53 mutations are, which suggests that they may be a hotspot or have some type of functional significance to warrant such a representation on this Lolliplots plot. So let's go ahead and start with the KM curve, and we can, I'm going to demonstrate how to make one of these curves. So you can load up your cBioPortal and you can be greeted to this page. And so I'm going to click on the studies that we want to take a look at. Perfect. And so we wanted to take a look at patients who have or do not have a TP53 mutation. So that's where we're going to right here. All right, now we're going to go to comparison and survival tab. Okay, here we are. And so you can see in red, this represents altered group. So we specifically selected for patients who had TP53 mutations. So this is all the altered patients. And also in blue is unaltered, so patients who have or do not have the TP53 mutation. And so what I typically do, I mostly focus on the patients on this side versus samples, mainly because some patients have multiple samples, which may, for this study, typically we want to look at individual patients. And so that's why we typically focus on this side of the panel. And some patients have multiple samples with and without TP53, so there's some overlap, and we want to exclude those patients within our analysis. And so to create these two separate groups, I just click on the altered group, and this is the TP53 mutant group. Submit. And we are going to do the same for the unaltered group and unaltered. So these group of patients are the wild type group, the WT for wild type. Submit. Okay, and we just are going to deselect these two groups and select our mutant and wild type groups. And we're going to go over here and click on survival. And we create the curves, the KM curves for comparing TP53 wild type versus mutant. And you can see, very nice addition to the KM curves generated on cBioPortal is that they do the statistics, the stats for you, and you can see this is a significant difference. And also one way you can observe this difference is just the immediate separation between the two curves. In addition, You can take a look at the bottom bars, which shows the number of samples. So they're pretty well represented, almost around 50% in both groups. You can see the number of events for overall survival. These events are death, and you can see the median overall survival for these two groups as well. In addition, if you want to take a look on the site, if you want to look at PFS, progression-free survival, disease-free survival, and there's other categories as well that you can also play around with. Also, I wanted to take a look and show you this clinical tab, and it also compares between groups certain characteristics that is seen and associated with each patient. So, for example, let's say I wanted to look at tumor mutation burden, and you can see comparing between these two groups, the tumor mutation burden, and it's pretty expected that patients with TP53 mutation would have a higher TMB than the wild type. So that's an additional thing that you can do when comparing groups, making KM curves, and also comparing certain characteristics that are inherent with the NGS. And one thing to add is that certain NGS panels may or may not have these certain characteristics as well, but TMB is well represented. So that's how to make the KM curves. And so let's move on to making these lollipop plots. And so I'm just going to make a new study. So similar thing, we're just going to scroll down and click on the studies that we want to take a look at. And once again, we're going to filter based on TP53 mutation. And you're just going to go to this mutations tab right over here. And the lollipop plot will just pop up. And there's ways in which you can also download the picture as well. And as you can see, the higher the bars, the greater, the more mutations, that specific mutation being represented. In addition, the colors of these, the ends of the bars, also describe what type of mutation that's being observed. And you can see in red the DNA binding domain, and over here you can see that's also being highlighted, the transactive motif and the tetramer as well. And so just to recap, we described how to make Kaplan-Meier curves. In addition, I want to highlight that you can also compare patients and their NGS characteristics, such as TMB. And lastly, going to the mutations tab, you can also create a lollipop plot as well. Thank you so much for watching, and I hope to see you soon on the next video. Thanks.